Welcome to HEXAPODCAST, brief discussions about insects and entomologists with your hosts, John Steyerman and Lee Dyer.
Hey, John Steyerman, how's it going?
Pretty good. How are you doing, Lee?
Great. I'm really excited to say we have another guest on our show, the one and only Laura Richards from UNR.
Can everybody hear me?
Yes.
All the listeners out there.
All two of them. Yeah. So let me just set the stage and John can interrupt me. We're going to talk about digestive systems.
Okay, this group will not be afraid to talk about insect guts, vomit, poop, or anything that happens from the mouth to the anus.
I'm not really going to say much at all. We're mostly going to listen to Laura. I'm just going to...
Oh, wait, I'm supposed to... Oh, I thought I'm just chiming in.
No, you're the special guest. Like, you're the
I'm
focus.
the special guest.
We're going to ask you some hard questions about...
Oh, okay.
Really about the midgut. Well, for you, we'll hope...
Yeah, because I mean, that's where a lot of action happens is in the midgut.
So, Laura, like, how did you get interested in working with insects in whatever regard you work with them?
At whatever level I do.
You know what I'm saying?
Yes. Well, first it actually started with undergrad. I was just super fascinated with insects and plants. I did my undergrad at UC Davis and worked with Don Strong over the summer. And I was coming up with all these ideas of how these leafhoppers penetrate these like grasses and blah, blah, blah. But then I went to grad school and I really actually wanted to do chemistry. But my PhD advisor told me that that was not going to be very fruitful for a PhD because I would spend all of my time trying to figure out what the compounds were. And at the end of the day, I wouldn't know what they would do. So I spent my whole time counting bugs in Panama. And so just spending a lot of time counting leaves, counting bugs, looking at patterns of, different trophic levels between gaps and understory. And then technology came a long way. And now we can really dive into some of the chemistry without being 100% of a natural products chemist. So when we think about the gut, I'm going to think a lot about how different compounds get in and out of it.
All right. So let me just go through the gut real quick. So typically when we introduce this system, we break it up into foregut, midgut, and hindgut. And then the foregut is basically just a tube to get everything to the midgut. But it starts with a true mouth. So you have the Siberian, you have a hypopharynx.
Wait, can I take a little issue here?
Sure.
I think the foregut, just a way to get stuff to the midgut, is a little bit glossing over the importance of the foregut.
John is interested in the foregut because he's a fly guy.
At this point, Dyer proceeds to drone on about salivary glands. Although technically not part of the foregut, the salivary glands and associated mouthparts are fascinating because of their diverse products, including silk, anticoagulants, and even digestive enzymes, especially in flies.
Well, actually, there are some butterflies that how they will regurgitate on plants that have pyrrolizidine alkaloids as they're like kind of wilting because they don't have the full mouth part as adults, right? So they kind of could put some saliva in there and then they slurp it up to get those compounds in to do some sequestration.
Exactly. This is what we talk about all the time when we talk about entomology, is that you have sort of this ancestral design or the orthopteroid plan that is modified in many, many ways. So if you start with the foregut just being this tube to get to the midgut where all the action is, then you can say, well, actually this and this and this, and that's what gives you insect diversity, I think. You've got the esophagus and the crop, and then in some insects you have a proventriculus, and then you've got a stomatal valve that leads you to the midgut. And in some insects, it's all about the crop. So if you're an adult ant, you don't really eat anything. You just store everything in the crop and you feed it to the larvae, and then they do all the digestion. So you have to have a big, huge crop.
This is where, like, when you poke a caterpillar and it regurgitates and spits that nasty stuff on you, that's all stored right there, right in the foregut.
Okay, you two are right. The foregut is amazing. Let's just move on to talk a little bit about the structure of the midgut and hindgut, and then Laura will tell us more about foregut, midgut, hindgut, whatever we want. You look at a diagram, they'll show gastric ceca as sort of the demarcation of the anterior portion of the midgut, and those aren't even present in caterpillars, which is what we study. But gastric ceca is where a lot of things get across the gut. So there's a bunch of trehalose in the hemolymph, So glucose goes across its concentration gradient at the gastric ceca in many insects. Also, in many insects, stretching the full length of the midgut is the peritrophic membrane, right?
Are we on the midgut now?
He's in the
Yeah, we're on
midgut,
the
you guys.
midgut. And then food
Yeah,
goes
he
through
jumped right
that,
in.
and then
Jumped right
that's
in.
what creates the countercurrent, so that food is going anterior to posterior, and then outside of the peritrophic membrane, it goes anterior again, and then it crosses all of these cells, the epithelium of the midgut. So that's it for the midgut. Laura's gonna talk more about that.
That's it.
And so the demarcation between the midgut and the hindgut is the Malpighian tubules, which I think are amazing. And then from there you have the anterior intestine, the rectum, and we'll talk a little bit more about the rectum, and then the anus, and then everything that comes out of the anus is actually pretty interesting too.
Here's a recap for those who couldn't hear what we were saying. The insect foregut is a dang important part. It is a cuticle-lined ectodermal region for ingestion, storage, mechanical processing, and maybe some regurgitation. The midgut is a non-cuticular endodermal region where most digestion and nutrient absorption occur. And the hindgut is marked by Malpighian tubules, and it is a cuticle-lined ectodermal region specialized for water and ion reabsorption and feces formation. Starman, want to add something important to this recap?
I just want to say one thing, so in a lot of ways, insects are really weird. They're very different from us. It's hard to relate to them. But the gut is, it's a gut, like in us, it has a lot of the same functions. But one of the cool things about insects that we'll probably talk about is aside from the midgut, like they molt out the lining of those guts, right? The foregut and the hindgut. So at first you're like, oh yeah, I can get that. It's like a gut like my gut. But then you have to imagine that they actually have to molt out their gut out their mouth and anus when they molt,
Yeah.
which is pretty awesome.
I hope it goes one way and not the other. If it was on me, I'd want it to go one way and not the other.
That's a great point, John. I always like to bring that up. And molting your foregut and hindgut, the exocuticle and the two-layer epicuticle that are just in the foregut and the hindgut, can be problematic. In termites, they have these microbial communities that help them or allow them to digest cellulose. And when they molt, they lose those, so they have to do this anal trophallaxis, sucking fluids out of the anus of a nest mate. This really happens. Is that what you teach, John?
Yeah, I mean, more or less, sure.
Well, what's different?
No. That's how I teach it. Yeah.
Okay.
I mean, the peritrophic membrane also kind of sheds, but often around the bolus of food moving through there, right? It's like continually regenerated.
Yeah.
Right?
Yep.
But it's kind of cool, right? It's kind of designed to like have this second layer of digestion that happens. It's kind of semi-protected from the bolus, right? Is my understanding that then you can have these enzymes in there that you're not going to slough off as the food keeps moving through. You can kind of keep them trapped in there.
Yep. But all this stuff is pretty variable. There's even a beetle that extrudes the peritrophic membrane to make a cocoon just before it pupates. So it extrudes it in the prepupal stage. But let's bug John about a midgut thing. How do tachinids typically get across a midgut into the hemolymph of something they want to parasitize?
Right. You're talking, everything comes back to tachinid flies.
Right.
So, yeah, there's some tachinid flies that lay these tiny, tiny eggs called microtype eggs on foliage that a caterpillar or other herbivore consumes that foliage, and somehow those eggs, they're really tiny and they have hard shells and they don't get crushed by the mandibles. And then there's some kind of enzymatic activation or something that happens in the midgut, and they hatch, and then they burrow typically through the midgut into the body cavity. And that's how they get into their hosts and then develop inside them and kill them.
Yeah, that's cool. So that's one of the ways that tachinids attack the caterpillars that we study. But the caterpillars we study are also plant feeders, and they have to deal with getting things across the midgut. So I wanted to ask Laura her quick summary of what happens in the midgut, or what happens in the foregut, midgut, and hindgut.
I'll tell you for a couple of things. So we generally have a big pattern that we see over and over again, depending on across lots of different plant taxa. The plants make these defensive compounds, and they're always like kind of packaged in this sort of mild mannered, configuration. So they often will have like a sugar attached to it. So something that's like really, really polar, right? So if you remember thinking about something that's, plants are made of water, it's going to come in, but now all of a sudden they're in the gut. And so whether you're talking about cardiac glycosides, iridoid glycosides, glucosinolates, cyanogenic glycosides, you'll hear this word glycoside over and over and over again. Even pyrrolizidine alkaloids, they come in a tertiary amine, and they have to be kind of converted. As soon as they hit the gut, there's all these enzymes that take that sugar off, and all of a sudden now it's super toxic. And now it has high permeability. It kind of can flow through that peritrophic membrane. It can now get into the epithelial. And depending on whether you're a specialist or a generalist, you might have these transporter proteins. So if you're a specialist and you're like, I want that thing intact, you might move it all with the sugar and keep it in there. I was actually reading up these couple of papers looking at these horseradish beetles. Have you guys heard about these beetles? They look at them up in Jena. They do a lot of work on them. And they actually found glucosinolate transporters in the foregut, which was like super groundbreaking. Like nobody expected transporters up there. Well, that was kind of a cool finding. And they also found that there's some transporters also in the Malpighian tubules, which we didn't talk a lot about, but those are like the kidneys. That's where a lot of detoxification happens. And so if you're a specialist and you don't want to lose this precious compound, you want to keep using it, they actually can some species can reabsorb it if they end up in those Malpighian tubules. And you're like, nope, I don't want that detoxified and excreted. I want to actually reabsorb it, which is kind of cool. It's like they're really like these compounds are really precious to them. But what's so funny is that all these compounds, they get into the gut, they kind of become more toxic. And then what do the insects do? They have to go through and they usually oxidize it. So they increase polarity once it gets into those hemolymph, right? They try to increase polarity to then spit it back out into the Malpighian tubules to excrete it. So it's this kind of this crazy trade-off between polar and non-polar, modifications that keep going on. And it's this repeated pattern we see over and over again, which is it's so cool to think about. So yeah, so that's like phase 1 detoxification is to use cytochrome P450 and oxidize that compound to increase polarity to try to get it to then be able to to excrete it through the Malpighian tubules. And then if that doesn't work, now you have to get costly, add a sugar, add some amino acids, and get crazy to try to excrete it. So that is the standard detox mode to get rid of these toxic compounds.
And that's the whole trick about the midgut is you want to get nutrients across. You don't want to let tachinid getting across the midgut. You don't want any kind of pathogen across the midgut, but you do want nutrients. And if you're using toxicity as a defense, that's diet derived, you got to get those across the midgut. And how does that work?
Well, they have these really not specifier proteins are what you could do for the glucosinolates that you get in the saliva, but they do have these specialized transporter membrane, these transmembrane enzymes that can actually transport those compounds unadulterated. So they might even keep that glucose on. They might have some special thing to like reduce. A lot of times specialists of like cyanogenic glycoside or a cardiac glycoside will actually have reduced the beta glucosidase enzymes that they have in their guts will be diminished, so that way it maintains that sugar on there, so then they can actually transport it completely whole. Or the sugar will be cleaved off. They will also, if you're a specialist, they will have these specific proteins to transport it across the midgut into the hemolymph. And that's where you then get these cool things, called this metabolic trapping, where then once it's now inside, you can then add that sugar on and now it won't leave again, right? So it's this cool way to like, you can put it in a cell and save it for your own purpose.
But But how do you kill that pesky tachinid that's in there trying to kill you?
Oh, well,
And so
that
get it into the hemolymph, and then if you put a sugar back on it, it's the tachinid is going to keep chomping away.
that's true. That is true. I have not thought deeply about. I just think they're swimming in this slurry of stuff. Sometimes they
Right.
put proteins on top. They will tag them. I've read about where they will take these compounds and they will kind of tag them to keep them inside, but they'll still be present in their more toxic state. But I don't know
Yeah.
exactly.
So I think it's safe to say that we know almost nothing about this process.
Yeah, okay, good. Because I'm like, I don't know. I haven't looked into that.
I mean, we don't
Like,
even know.
okay, I thought these weren't supposed to be gotcha questions. I thought we were supposed to have fun.
You say that we don't think about, like, if I were to say, give me a list of things that we know are actually sequestered, it'd be a pretty short list, wouldn't it? We don't even know
Oh
what is
yeah.
sequestered.
Well, no, it's super short. And also things that are sequestered, you have to show that it was sequestered with intention, right? It just can't be passive diffusion across the epithelium and then, just happens to be in the hemolymph because they couldn't do anything else about it. So,
Well, yeah, that's what Clive Jones called this casual sequestration. And I think mostly what he was talking about is the fact is that if you eat nasty stuff, you're nasty even if it's not part of your tissues. So anything that's in your gut is going to be eaten. Unless you're one of those wasps, there's some paper wasps that will grab a caterpillar and they'll dissect out its gut and they'll throw the gut away and then they'll eat the rest. But yeah, I think that's the problem is that we have these great stories, and they're just stories that are well studied, like with monarchs and
yeah, the very
monarchs.
limited systems that we actually know what is really going on.
Can I just like say
Yeah,
one more thing about tachinid flies in the gut?
yeah, of course.
Okay.
I might just cut it, but go ahead. So,
No, one like kind of cool thing is the most prolificous tachinid that we know that attacks the most different species, all kinds of families of leps and like non-leps and sawflies and stuff, Compsognathus cancellata. Anyway, they actually showed that it doesn't develop in the hemocoel or like it doesn't develop in the body cavity. It actually develops in the midgut between like the peritrophic membrane and like the epithelium
Oh, that's
so
smart.
it's yeah,
cool. Yeah.
which kind of protects it from the immune system, but I don't
It
know
protects
how
it,
it.
yeah. And it would get a lot of like breakdown, like essentially the caterpillar would be digesting everything. So if it got in there, it'd be kind of a smaller, sort of amino acids, like really digestible, sort of nutritious space to be in.
It's kind of the sweet spot because you know how a lot of caterpillars regurgitate. They basically just they'll make a crude plant extract, and that crude plant extract is pretty toxic for most things. So that's what they're regurgitating. But the tachinids are after a lot of things have been, enzymes from the salivary glands, enzymes that are right at the beginning of the inside of the peritrophic membrane. They're getting food that's pretty good and safe, right?
Yeah.
If they're
Mhm.
outside of the peritrophic membrane.
Yeah, and I think that well, I don't know, I mean, they are getting exposed to whatever phytochemicals are being ingested. There's also an issue of low oxygen, and I think they like basically poke a hole and drag a trachea from the body cavity into
The
the
snorkel.
midgut. Yeah, basically make a snorkel. Yeah.
Yeah, that's cool. Takinas are pretty cool.
Well, I mean, I think though, like when you think of sequestration, you think of or using that as a defense, you think of an accumulation. So concentration would be really high in the hemolymph. So regardless, like in there, the concentration is probably lower than if it was actively accumulating it into the hemolymph itself.
Yeah.
Good point.
Okay, so we have to move to the hindgut, and I want to ask Laura
Oh,
about her obsession with caterpillar poop.
I do like
Laura
poop.
is making this technician in Costa Rica collect a whole bunch of caterpillar poop, and I want her to
Yeah. Well, I
explain
think it's
why.
kind of interesting to know what actually makes it through. Like, so we have just described this whole gauntlet that this like food bolus is having to battle as it goes through an insect, right? It's like there's salivary things, you're chewing it up, there's like diffusion. And so figuring out what actually ends up making it through in the end is kind of interesting and can give you a lot of details. I'm hoping when we do the chemistry, but we also know it's going to be a little bit challenging, right? So you do have the Malpighian tubules feeding into what's coming out. So things can be altered. So even if things, you like to think of it as a tube going, what goes in and what's coming out is the unadulterated version, but you still have all of this mixture of these detox byproducts, right? These things that they've done to protect themselves. So I don't know. I feel like we can learn a lot from poop, and you can get more poop.
Well, mostly potty humor, poop, vomit, that kind of thing. Really? That is like the lowest form of comedy.
To analyze than maybe you can in a small caterpillar. Maybe it can just process a bunch of stuff and give you some insight as far as, biomass to extract. So
Caterpillars are amazing synthetic chemists. Whenever I talk about Miriam Rothschild work with Arctia caja, which was an archaean that ate cannabis sativa and sequestered really high levels of THC, students would always say, oh, well, can you smoke it? And I think the levels of THC were the highest in the frass. And I would always say, well, you have to
oh
smoke.
yeah, smoke some poop.
Now, I was in that shit, man. I never had no dope like that before in my life, man. That's the heaviest shit I ever smoked, man. I mean, I smoked a lot of shit before, man.
Okay, let's finish up with one last question for each of you. If you had $1 million.
$1 million.
Okay, $1 billion to do some research on the gut, what would you focus on? What do we really need to know about the gut? And I'll start with John.
Well, I don't know, man. I don't work on gut stuff. No, but I think all this, the diverse mechanisms by which plant-feeding insects, deal with chemical defenses in plants. I mean, I think that's a pretty big one. I mean, most insects are feed on plants, right? And there's all kinds of specialization, and there's lots going on there. So even though I want to talk about flies, I think really herbivorous insects. I think there's probably a lot more interesting things going on with plant-feeding insects.
Yeah, I'd be interested in seeing like just sort of like broader surveys of different non-model systems and seeing what is passively being accumulated in the hemolymph versus actively, like just kind of looking at different patterns like that would be really interesting. I did feel like we did miss something really important because I did think about the anus a bit, and
Oh, excellent. Let's do the anus.
there are some fruit flies.
Nothing stopping you.
There we
Go
go.
ahead.
Anyways,
Give us
there
the
are
anus.
these fruit flies that get eugenol from their plant, right? They sequester it, which is it's a monoterpene, so it's super volatile, which is kind of cool. It has to pass all the way through their gut. They store it down there to then use it as a pheromone for sex.
They
Like
store it
to
down
attract
there?
the ladies. Yeah, their
What do you mean
rectum.
by down there?
Down there,
What? Okay.
down there. No, in the out of the rectum, because they consume it, so it has to come out, and they have these special. They release it in their anus
Okay.
to attract mates.
The seventh planet from the sun is called Uranus, known as a gas giant.
I don't get it. It's just science, guys.
There's a whole related field of fecal ecology, like Martha
Yeah.
Rice stuff, where there's poop shooters that shoot out the poop out their butt, and how where they put their poop, and
Yeah, they could wear it. They might wear it.
you can wear it.
Fecology.
Ecology.
That's trying to make it sound fancy.
So Beto, when he looks for caterpillars in Costa Rica, he looks for frass on the ground if he's looking for big caterpillars. And the big, huge silk moth caterpillars and hornworm caterpillars, their frass is big and recognizable, and they're usually up in the trees. So if you see their frass on the ground, they're up there. Because of that, a lot of visual predators find their prey by smelling the frass or seeing the frass. And so, yeah, a lot of caterpillars shoot it pretty far. I think we recorded a noctuid shooting at about 1.5 meters. Really small.
Pretty good. That's impressive.
And the poops are cool looking sometimes. You got it's like you use the Play-Doh
Oh, they're so
squisher
cool.
thing. Like
Their
you have
shape is so cool. Yeah.
But my
Yeah.
favorite party trick that I've seen from a caterpillar is that they actually grab their poop as it's coming out of the anus and then they flick it. So they grab it with their mouth because they don't have any hands, right? So they grab it with their mouth as it's
Good.
and then they
Glad
flick
you
it. They
specified
throw it
that.
pretty hard. So that's a party trick that I haven't been able to replicate.
Yeah, I would suggest not trying.
All right. Thanks, everyone.
Okay.
See you later.
Bye, guys. Good to see you.
Good to see you again, Laura.
Yeah.
Thanks again to all you insect lovers for listening to HEXAPODCAST. We will see you next time for a discussion of insect breathing entitled Tracheal Tales. Really impressive stuff.
On the wheel right by your side. Hold on to the end of the line. Lay with you while the night is still. Proves you haven't lost your will. Soon the road will be clear. The days only go